Enzymes. Enzymes are proteinaceous substances which have the catalytic property. Enzymes are the proteinaceous substance, means all enzymes are protein in nature which have catalytic property, means the enzymes does not initiate any reaction but they increase the reaction rate. They increase the reaction rate. It is the catalytic property, okay, of enzymes. So enzymes are the proteinaceous substance which have catalytic property. Then the term enzyme first coined by German scientist Kuhn, German scientist Kuhn in 1877. Okay. Then some characteristics of enzymes. At first I told that enzymes are protein in nature. So first point is the enzyme. All enzymes are protein in nature. Then next point, they are colloidal substance. What is the colloidal condition? Colloidal condition is that in which condition any substances does not dissolve in a solution, does not dissolve, and also does not precipitate in a solution. That state of any substance which are present in a medium, means liquid medium, is called colloidal state. Then all enzymes which are present inside a cell, they present in colloidal state. Okay. Then third, they do not start a reaction but increase the reaction rate, means it is the catalytic property of enzyme, or enzyme have the catalytic property. They do not start a reaction but increase the reaction rate. Then they are unchangeable. Unchangeable means after a reaction the structure of enzyme does not change. The structure of the enzyme will not change. For that, enzyme are unchangeable in nature. Then reversible. Reversible means if any enzyme takes part in a reaction, then they convert the substrate into the product, and also they convert the product into the substrate again. It is the reversibility of an enzyme. As example, glucose 6-phosphate glucose 6-phosphate are converted into fructose 6-phosphate by presence of hexokinase enzyme, and also the hexokinase enzyme also helps in conversion of fructose 6-phosphate into glucose 6-phosphate again. It is the reversibility. Specificity. All enzyme has a specificity, means all enzyme has a specific substrate on which they will react. For that, it is the specificity. For example, the enzyme amylase which are present in our saliva, called salivary amylase or ptyalin, it convert only the starch into maltose, means starch is the substrate of the amylase enzyme. Okay, it is the specificity of an enzyme. Then classification. We can classify the enzyme depending on many purpose, such as first, depending on the chemical nature of an enzyme. Depending on the chemical nature of an enzyme, we can classify the enzyme into two groups, that is, these are the simple enzyme and conjugated enzyme. Simple enzyme means if the enzyme formed by protein only, then this enzyme is called simple enzyme, such as pepsin, amylase, etc. Then conjugated enzyme also known as holoenzyme, means it has the protein part and also some non-protein part. The enzyme part of the holoenzyme or conjugated enzyme is called apoenzyme, and the non-protein part of the holoenzyme or conjugated enzyme is called cofactor. We can also classify the cofactor into three groups, such as coenzyme, prosthetic group, and metal ions. First, coenzyme. Coenzyme are those substances Coenzymes are those organic substances which are attached with the apoenzyme with the apoenzyme. These are known as coenzymes, such as NAD, NADP, FAD, riboflavin, etc. prosthetic group. Some some organic substances which are attached with the apoenzyme by a close bond or tightly attached with the apoenzyme. The organic substances which are tightly attached with the apoenzyme is called prosthetic group, such as heme, biotin, etc. Then metal ions. Some metal ions also takes part in the reaction of an enzyme. Some metal ions which are helps in the activation of the enzyme, such as molybdenum. It takes part, or it helps the nitrogenase enzyme at the time of nitrogen fixation. Some other are copper, manganese, zinc, etc. Those are the metal ions. Now we can also classify the enzyme depending on their function. Depending on the function of an enzyme, we can classify the enzyme into 6 groups. First group is oxidoreductase, means those enzyme which helps in oxidation and reduction of the substrate. As example, oxidase, dehydrogenase, those are the oxidoreductase enzyme. Then transferase transferase enzyme helps in transfer of a specific group in between two substrate. It helps the transfer of specific group in between two substances. As example, transaminase, which transfer the amine group from glutamic acid to another substrate for forming different types of amino acid in cells. Then hydrolase. Hydrolase means water. Water takes part in the reaction of the enzyme. Those enzymes are called hydrolase. Hydrolase enzymes break down the substrate. Hydrolase enzymes which break down the substrate in presence of water, such as amylase. Okay. Then lyase, breakdown of the substrate without the water. It break down the substance, means it work as like hydrolase enzyme, but hydrolase enzyme function in presence. Hydrolase enzyme convert the substrate at the presence of water, but lyase enzyme does not need water for breakdown of the substrate. As example, aldolase. Aldolase enzyme break down the fructose 1,6 bisphosphate into two 3-carbonaceous compound, such as 3-PGLD, means 3-phosphoglyceraldehyde, and dihydroxyacetone phosphate. Then ligase. Ligase means the enzyme which joins to substrate is called ligase. As example, RUBP carboxylase. RUBP carboxylase joins the RUBP and carbon dioxide to form two molecule of phosphoglyceric acid. Then isomerase. Isomerase enzymes are those enzymes which changes the substrate into its isomer. It changes the substrate into its isomer. As example, phosphohexoisomerase. It converts the glucose 6-phosphate into fructose 6-phosphate. Fructose 6-phosphate is the isomer of the glucose 6-phosphate. Another example of isomerase helps in the conversion of ribulose 5-phosphate to ribulose 1,5 bisphosphate. Ribulose 5-phosphate to ribulose 1,5 sorry ribose 5-phosphate to ribulose 5-phosphate. Ribose 5-phosphate to ribulose 5-phosphate. This conversion also occurs in presence of isomerase enzyme. Then some other enzyme. First, proenzymes. Proenzyme means the precursor or primitive form of the enzyme of an enzyme is called proenzyme. Okay, proenzyme means those substances which are converted into the enzyme is called proenzyme, such as pepsinogen. Pepsinogens are secreted from the gastric gland of our stomach. In presence of HCl, they convert into pepsin. Pepsin is the main enzyme, but pepsinogen is the precursor of the pepsin enzyme. For that, pepsinogen is the proenzyme of pepsin enzyme. Then isoenzyme. Isoenzyme means those enzymes which have similar structure but play different role in a cell. Those which have the same chemical structure but have but they have different function in a cell is called isoenzyme. As example, lactose dehydrogenase. Inside a cell, lactose dehydrogenase has five isoenzymes. Then abenzymes. Abenzymes means if any antibiotic act as an enzyme. If any antibiotic act as an enzyme, then the antibiotic is called abenzyme. Then ribozyme. Ribozyme means if any RNA act as enzyme is called ribozyme. It takes part in the translation. Translation means the process of formation of protein. The process of formation of protein from the mRNA. Then allosteric enzyme. All enzyme has an active site. All enzyme has an active site. But if any enzyme contains more than one active site, those enzymes are called allosteric enzyme. Okay. Then one site is called active site, in which the substrate gets attached, and another site is called allosteric site, where any other substances will attach with the enzyme. Those substances which are attached with the allosteric site of an enzyme are also classified into two groups. These are inducer and inhibitor. Inducer means those substances which are attached with the allosteric site of an enzyme. For that attachment, it increases the enzyme action. Then it's called inducer, or it helps in the enzymatic action. For that it's called inducer. And another substance is inhibitor, which if those substances are attached with the allosteric site of an enzyme, then it inhibits the enzyme reaction. Okay, so those substances are called inhibitor. Then endoenzyme and exoenzyme. Endoenzyme, if any enzymatic reaction occurs inside a cell. Means if any enzyme functions inside the cell. Okay, then it's called endoenzyme, such as RBP carboxylase, hexokinase, etc. And exoenzyme means the enzymatic reaction. If the enzymatic reaction occurs outside the cell, then it is called exoenzymes, such as amylase, pepsin. Okay, those are the exoenzymes. Then mechanism of enzymatic reaction or enzymatic action. How does the enzyme act on the substrate? At first, enzymes are attached with the substrate. To form a complex, which are called enzyme substrate complex or ES complex. Then when the ES complex are formed, then the enzyme does not change, but substrate is converted into product. So after forming of the enzyme substrate complex, then the enzyme remain unchanged and substrate will change into its product. So it is the basic action of an enzyme. You remember it, the enzyme lowers the activation energy. First, what is the activation energy? Most reaction do not start automatically. Any reaction do not start automatically because the reaction have an energy barrier. To become reactive, which may be due to hydrogen bonding. For the presence of hydrogen bonding, all reactions are needed, or all reaction have a energy barrier. If we externally supply the energy, if we externally supply the energy, then the barrier will break down. Then the barrier will break down, and the energy which which are externally supplied by us is called activation energy. So we can also define like it, the external supply of the energy needed to overcome the barrier, overcome the energy barrier for a reaction is called activation energy. So the external supply of the energy needed to overcome the energy barrier of a reaction is known as activation energy. And the enzymes lowers the activation energy by following way: by taking substrate out of the solution state, state. Then by forming weak bond between the substrate and enzyme, or by binding, or by bringing the substrate close to one another in the region of active site. By these three steps, by any one steps of those which are given here, any one of them okay, any one of them lowers the activation energy. Look here, that is the x line, and that is the y axis. It is the x axis, and that is the y axis. X axis determines the reaction rate, and y axis determines the potential energy which are needed for an chemical reaction. If we do not use any external energy, then a chemical reaction need those amount of energy for activation a chemical reaction. But if we use an energy for a chemical reaction, then it needs only this amount of activation energy. Okay, so this is needed for the chemical reaction without enzyme, and this much only needed as activation energy for the presence of an enzyme means enzymatic reaction. So the enzymes lowers the activation energy of a chemical reaction. Then the hypothesis for enzyme reaction or enzyme action. First, we discuss about the lock and key hypothesis proposed by Fischer in 1894. First, lock and key hypothesis. In lock and key hypothesis, scientists told that the both enzyme and substrate molecule have a specific geometrical shape. Both enzymes and substrate molecules have specific geometrical shapes. In the region of an active site, the surface configuration of the enzyme is such as to allow the particular substrate molecule to be held over. Means, according to lock and key hypothesis, all enzyme have a specific activation site which are similar to a specific substrate. For that, those substrates are easily attached with the active site of the enzyme, and they form the ES complex. Then enzyme remain unchanged and the substrate break down into products. That is the lock and key hypothesis. According to this theory, it is similar to the system of a lock and key. Just as a lock can be opened by its specific key, a substrate molecule can be acted upon by a particular enzyme. It is similar to the system of lock and key. Just as a lock can be opened by this specific key, a substrate molecule can be acted upon by a particular enzyme. When the substrate come in contact with the active site of the enzyme, the substrate molecule form a enzyme substrate complex, and then the substrate convert into product and the enzyme remain unchanged. It is the lock and key hypothesis. After it, another hypothesis is also applied in the enzyme reaction, that is the induced fit hypothesis or induced fit model. Induced fit hypothesis proposed by Coastland in 1959. According to this theory, the active site of an enzyme contains two groups, buttressing group and catalytic group. According to this hypothesis, all enzyme contains two groups, buttressing group and catalytic group. The buttressing group is meant for supporting the substrate. The catalytic group is able to weaken the bond of reactant by electrophilic and nucleophilic forces. The catalytic group is able to weaken the bond of reactant or substrate by electrophilic and nucleophilic forces. As soon as the substrate comes in contact with the buttressing group, the active site of the enzyme undergoes conformational changes so as to bring the catalytic group opposite to the substrate. Catalytic group helps in bringing about the chemical reaction and substrate is converted into product. Means all enzyme has two group, buttressing group and catalytic group. The active site, as simple way, according to the induced fit hypothesis, the structure of the active site of an enzyme is not similar to a substrate. At first, substrate come contact of that activation site of an enzyme. Then the active site will change according to its substrate. Means that is the buttressing group of an enzyme where it holds the substrate. And according to this theory, then the catalytic group takes part for changing the shape of the activation site. Then easily the ES complex is formed or ES complex will formed, and then the enzyme remain unchanged and the substrate converted into the product. It is the induced fit hypothesis proposed by Coastland in 1959. Okay. Then I come at the point factors affecting the enzyme action. First factor is concentration of enzyme. Biochemical reaction rises with the increasing of the enzyme concentration. It means that if we increasing the enzyme concentration in a chemical reaction, then the rate of reaction will increase up to a specific point, and this point is called saturation point. Beyond the saturation point, the reaction rate will be decreases. Okay, so if we increases the enzyme concentration with a certain level, the enzymatic reaction will increases for a certain level, and then the reaction rate will decreases. Then the next point is concentration of substrate. If we increasing the substrate concentration gradually increases the velocity of enzyme reaction up to a certain limit. Means as previous, if we increase the substrate concentration in a chemical reaction, then also it increases the enzymatic reaction rate up to a certain limit. After that, the reaction rate will decreases. Michaelis-Menten constant. Michaelis-Menten constant, it is the substrate concentration. It is the substrate concentration to produce half maximum velocity in an enzymatic reaction. Means the substrate concentration which produce the half maximum velocity in an enzyme reaction is called Michaelis-Menten constant. Remember that low Km value, Km means Michaelis-Menten constant, low Km value indicates a strong affinity between enzyme and substrate, and the high Km value indicates the weak affinity between enzyme and substrate. We can express by the following equation. We can express the Michaelis-Menten constant in following equation. Equation is that V is equal to Vmax S by Km plus S, where V is the measured velocity, Vmax is the maximum velocity of the enzyme, S means substrate concentration, and Km is the Michaelis-Menten constant. If you look at that graph, look here, the x-axis determines the substrate concentration and the y-axis determines the velocity of the enzyme. If we increases the substrate concentration, that line, if we increase the substrate concentration, then it increases at the highest point. It is the peak point of enzymatic reaction. It is called the Vmax. Then the half Vmax. Means when the velocity of enzymatic reaction reached at the half of Vmax, in which substrate concentration, then this substrate concentration is called Km value. Okay. Then the third factor is temperature. The temperature at which an enzyme shows its highest activity is called optimum temperature for that particular enzyme. The temperature at which an enzyme shows its highest activity is called optimum temperature. It is the for most of the enzyme is between 25 degrees centigrade to 35 degrees centigrade. Enzyme activity decreases above and below the optimum temperature. If we decreases the temperature from 25 degrees centigrade or increases the temperature from the 35 degrees centigrade, then the enzyme activity will decreases. Okay. Then the pH value. Each enzyme has a optimum pH, same as temperature. Each enzyme has a optimum pH. Below and above this pH, the enzyme activity decreases. Okay, or the enzyme does not react. As example, pepsin. For the action of pepsin enzyme in our stomach, it needed 1.5 pH. If we increases or decreases the pH level, means if we decreases the pH into 0.5 or increases the pH into 4, then the pepsin does not break down the protein. Okay, so all enzyme has a specific pH in which they can react or they will react. Then the product concentration. If the product concentration is increases in a medium, then the enzymatic reaction will be decreases. Okay. Then another important thing for enzyme, that is the inhibition of enzyme action. inhibition of enzyme action. Reduction or stoppage. Reduction or stoppage of enzyme activity due to the presence of adverse condition or chemicals is called inhibition. Reduction or stoppage of enzyme activity due to presence of adverse condition or certain chemicals is called inhibition. Inhibition, we can classify the inhibition process into two groups. These are the reversible and irreversible. Reversible inhibition means where the enzyme inhibition can be reversed. Means if any enzymatic reaction is inhibited, then it must be converted into its function again. It is called reversible inhibition. And irreversible inhibition means the inhibitor does not reverse. Means if any enzyme gets inactivated or inhibited, then it does not come back into its normal states or normal reactions. This process is called irreversible inhibition. Reversible inhibition we can also classify it into two groups: competitive inhibition and non-competitive inhibition. Look here. Competitive inhibition means if any enzyme, if the structure of an substrate and the structure of the inhibitor is same, then they both are want to attach with the active site. For that, a competition will occur in between substrate and inhibitor. It is called competitive inhibition. Okay. If the structure of any substrate is similar to its inhibitor, then they also compete. They also compete for the attachment with the activation site of an enzyme. It is called the competitive inhibition. And non-competitive inhibition is that where the structure of the substrate and inhibitor are differ. The chemical structure of the substrate and the inhibitor will differ. For that, the substrate get attached with its activate site and the inhibitor attached with the allosteric site. For that, no competition will form in between the substrate and inhibitor. It is called the non-competitive inhibition. Why it is called inhibition? If the inhibitor gets attached with the allosteric site of an enzyme, then it prevents the attachment of the substrate with the activation site of enzyme. For that, it is called non-competitive inhibition. Then another inhibition process is here, that is feedback inhibition. If the last product of a series of enzymatic reaction inhibit the first reaction of that series. If the last product of a series of enzymatic reaction inhibit the first reaction of that series is called feedback inhibition. As example, a substrate, enzyme 1, enzyme 2, enzyme 3, enzyme 4 and enzyme 5 takes part in a series of chemical reaction, and the substrates are A, B, C, D and E. Then the substrate A converts into B. The substrate A converts into B in presence of E1 enzyme. Then the B substrate is converted into C in presence of E2 enzyme. Then the C substrate converts into D in presence of E3 enzymes. The substrate D is also converted into E by presence of E4 enzyme. And the substrate E is converted into F in presence of E5 enzyme. Then the last product is the F. If now the F product inhibited the action of E1 enzyme, if the last product means F inhibit the action of E1 enzyme, then the first reaction does not occur and total the series will stop. It is called feedback inhibition. Okay, thank you.